Hi everybody. So we are going to focus on the next subject matter for this topic. And so we left off with eukaryotic DNA and looking at how it's able to wind up extra tight right before cell division. Now what it looks like when it wraps up really tightly is it forms what's known as chromosomes. These are highly condensed pieces of DNA. And you can see that you have two sides because you have now replicated those chromosomes. And so those replicated chromosomes are the sister chromatids, and they're connected by what's known as a centromere. This centromere is where they're going to be split in half later. And so they're extremely compact, and it allows this genomic material to be moved around really easy and then divided into two nuclear cells later. Now, when we look at them, the chromosomes become visible during the actual cell division, which is called mitosis. Before replication, the DNA is visible as chromatin. So this kind of gives you an idea of what it looks like. It's kind of this dark, you can't see the individual lines. And so this is unwound pieces of DNA. And during the beginning or subphase to division, there's going to be a place where this DNA replication happens, and so you're going to have basically this one strand here is going to replicate, and then we're going to have these two pieces of DNA through semi-conservative replication that is joined by that centromere. And so these have the same gene types on them. They may not be the same form of genes, but they do have the same gene types. And they start looking at these lines when you visualize them underneath a microscope in cells or organisms. And so we are going to focus on mitosis. And mitosis is going to allow us to generate an exact copy of the cell. One pass of mitosis takes one cell and duplicates it into two cells. At the end of mitosis, the two new cells, called daughter cells, have the exact same DNA as the parent cell does. And so you can look below, and there's this really nice cell animation for the introduction to what we know as the cell cycle. Mitosis is part of a larger cycle called the cell cycle. It's going to have a preparatory phase called interphase that helps the cell get ready to divide, which is called mitosis. So you do not need to take any notes on this. You're just going to watch it. You can find the link below in the description, and that'll get us ready for our discussion. So the cell cycle is a repeated series of events. It lasts from the beginning of one cell division to the beginning of the next. Includes the time when cells are divided, which is the mitosis, and the time when they're not dividing and getting ready for division, called interphase. As you can see, this process right here is the preparatory phase. And this right here is going to be when they divide the nuclear material. Cytokinesis, which is the last part you can see, looks like cytoplasm, and kinetics, right, is when you take the cytoplasm and you divide it there. So those are some main stages. Now, interphase, as you can see here, is not part of mitosis. Mitosis is actually when you can see it and divide. So if you're taking like a 12-hour and looking at this, 30 minutes would be mitosis, and 11.5 hours would be interphase. And this makes total sense to me if you think about moving. If you're moving from one home to the next, you're going to have the phase for which you're preparing for the move. You're cleaning your house, you're boxing everything up, you're making plans. You may be even purchasing additional furniture to put in your new house. So that is what I would consider to be kind of like interphase. People outside your home can't see that it's happening, but there's a lot of work going on, and it takes a whole lot of time because if you think about it, which one takes more time? getting ready and prepping for the move or the move itself. And if you've ever moved, you know, prepping for the move can take months, weeks, and the move itself can take a couple days if you have helpers, right? So that cell division is equal to the actual move, because at that point, people can see you're taking your stuff from one place to the next, where here you're taking your chromosomes from one place to the next and everybody can see it. So that's kind of the example I can give for what's happening in the cell cycle. So again, interphase, the cell's not dividing, but a lot of things are being carried out during this time. And so if nothing's happening and it's not doing anything, it can be called G0. And I'm thinking, not anything is happening, right? And that's kind of a suspensatory phase where nothing is really being carried out. They're not getting ready for division because you're not moving all the time. You're moving when it's important and you have to do it. So there are a lot of times that the cells are just living their life, not getting ready to divide. But when they do, that's when this G1 phase happens. This is when you have normal cell function, but also cell growth, right? You're making, because if you're going to divide the cells and they're going to be the same size, you need more of the cell membrane. If you want there to be just as many mitochondria in one cell as the other, you're making more mitochondria. You're doing all that activity. And then you're going to go to the next phase, which is S. So you can think synthesis. And if you're synthesizing something, you're making something new. And this is where the DNA replication that we talked about previously comes into place. So this is when we're making our 2 strands of DNA. So just to let you know as a side note, the number of chromosomes that we normally have in each one of our cells is 46. So in this point, we're doubling them to 92. And then you have G2. The G phases stand for gap. You can think there's a gap between these phases. And so you'll get additional growth if you haven't made enough during G1 or your cell realizes you need more, that'll happen. And this is the point where they're preparing and making enzymes and everything for division. And so cell division happens with mitosis. And that is the next phase that we're going to talk about. So just as a reminder, though, that G1, again, is the growth and function phase. So this is where protein synthesis is happening. This is where all the additional organelles or phospholipids are being made. But the cell is generally functioning normally. If it's getting ready to divide, it will move on to S phase. It cannot and will not stay in G1 phase forever. If nothing is happening, it's going to be in G0. The S phase is where DNA replicates and copies its entire genome, every single chromosomes. Now it's committed to divide, so there's no going back at this point. During G2, this is where they're producing specialized proteins that are necessary for mitosis. And then we're going to get into what's called the M phase, which is mitosis. So cells will leave G2 and enter mitosis. And this is where we're going to divide the nucleus. And then cytokinesis, remember, is dividing the cell itself or the cytoplasm. So we have 5 phases in mitosis. And you can remember them in PMAT or pass me another taco, because you know I love food. And so, you know, we're in Texas, so we have the best Tex-Mex tacos ever. And so PMAT stands for prophase, metaphase, anaphase, and telophase. And these are very distinct phases that you can see underneath the microscope. And then all of that, telophase, late anaphase, early telophase, is when you start seeing cytokinesis. And so you're going to start with one cell, and you're going to produce 2 daughter cells. In this example, you see there are 4 chromosomes, and so you will be ending with 4 chromosomes at the end. Now, on here, though, when we're talking about an interphase, that you can see here that each one of these chromosomes are basically connected here but remember there are chromatids there. And so essentially what's happening when they're saying this, so this is why I don't want you to get confused, is there are 4 duplicated chromosomes here, and then there are regular just 4 non-duplicated. Do you see how this is? So technically, you can go from having 4 chromosomes here, but they're duplicated. So that's almost equivalent to 8. So in prophase, this is where you're going to see the DNA is going to condense. Now, not just that, spindle fibers are going to form from the centrioles, which you can see here, and those centrioles go to opposite sides. So that's something additional happening. And if you look here, you'll notice that the nucleus is disappearing. So those are the main things that are happening in prophase. And at this point, the nucleus is going to disappear because that's the house the DNA lives in. And we want to be able to move all the chromosomes to line up so we can split them later. So in this phase, you'll start seeing that the chromosomes are going to start moving too. And this shows you a perfect example of basically what it looks underneath the microscope. So you can start seeing here that they're starting to condense, and you'll start seeing lines. And here, you'll start seeing right there, those lines that are coming that go like that, those are what are called the spindle fibers. So, and you can see here that those chromosomes are not yet lined up in the middle, but they're not in a small circle like here. So that's what you can consider to be prophase, early and late. The next thing we're going to see is metaphase. This is where they're the most condensed. So later we're going to look and be able, for instance, if you go in and you want to see and have your potential or your embryo tested for genetic abnormalities like Down syndrome, where you can have extra chromosomes, they form what's called the karyotype. And I'm going to talk about that later in a different sequence, or when we get to both meiosis and when we get to talking about genetics. But they use the chromosomes that are in metaphase here because they are the most condensed. And think, they're about to be separated or pulled away, so you need them to be super condensed so they're strong so the DNA doesn't unravel. So they're going to meet at the mid plate here. And you can think middle and M. So here you can see them all kind of lining up in the middle, and remember the cells are small, so you know you have that, and that's the reason why they look so large there, but they're in the middle basically. The next thing is they're going to go away from each other. So this is when the chromosome splits into 2 sister chromatids, and those chromatids get pulled apart by the spindle fibers and go into the opposite poles of the cells. And so you can see here where they're basically moving away from each other. And then you're going to have telophase. And telophase, I just think basically the opposite of prophase. So what happens here? Well, the nuclear envelope comes back, the chromosomes uncondense, the centrioles are together on one side, and the spindle fibers are gone. Now, during this process, so you're going to see a new nucleus forming. And then, basically, you're going to get cytokinesis that happens at the end. And that's when the cytoplasm is going to separate, and the 2 nuclei are going to be into different daughter cells, and they separate. So you can start seeing, and how you can tell here is you'll start seeing here, there's a line. And that is being formed to be the new wall for each cell. This is a plant cell, right? And so you'll notice that in this one, this is an animal, and I can tell because there's a contractile ring. And essentially we're going to have them divide, and they're going to, initially they can go into G0 if they're not doing anything else, but if not, they can go back into G1. Now, cytokinesis in animal cells produces what's called cleavage furrow, and it looks like cleavage there. And so they have a contractile ring, which works like a drawstring in your pants. So they're going to contract and get smaller and smaller until the two cut off. And so that's a lot of times you can tell that you're in telophase here because you can distinctly see a cleavage furrow. In plant cells, remember, they have a cell wall. So they cannot do what animal cells do. Animal cells don't have a cell wall, so you have that fluid cell membrane there. But we know that with plant cells, they're really strong and structured. And so we can't do the same process. So here we're going to be forming a cell plate. What happens there? Well, you have all these vesicles. And these vesicles have membrane and cell wall in them. And so they get recruited to the middle. And they're going to keep coming together and growing, you can see it here, until you have two new cells. So they're made up of like cellulose and other polysaccharides and proteins, things that are similar to what you find in your cell membrane and cell wall in plants. So the cell cycle is extremely controlled so that you have checkpoints throughout the process that regulates if the next step happens. So these checkpoints will assure that DNA replication has happened, the DNA is not damaged, that chromosomes are lining up and separating properly, and if they're not, it won't continue on to the next step. If they continue on to the next step, this is where dysfunction can happen. And things like Down syndrome or trisomy 21 can occur. Or actually, that's true, but that's a meiosis. I'm sorry. Something like this where they're not is where cancer will happen. Too many different things happening right now. So here is a review of mitosis. So you will want to take notes for this, and you can see the video below in the description, and this will help you to kind of overview and be able to see what it actually looks like. So yeah, if these checkpoints go out of control, then that is basically out of control cell division, and cancer can happen. So you can get tumors from uncontrolled cell division. Now, benign tumors mean that there are these kind of groups of cells together, but they are formed within a capsule, and they're easy to remove with surgery, and they are not causing problems as what's called metastasizing. So a malignant tumor basically have cells that can seed new tumors throughout the body and spread. So benign tumors aren't spreading. They're contained with connective tissue. And malignant cells can seed. So you can see that they don't have any containment there. So what happens is they can be taken up by the lymphatic system or even in blood vessels. And because those are highways, superhighways to your whole body, then those cells can move to other places where they create new tumors. And the problem with these cells is that they don't function correctly. Even say, for instance, if you have, for instance, a cell that's a cancer cell that starts in your liver, it's not working like all the other liver cells. So it's basically taking up space. Other cells are dying because these are taking up space, and they're not performing the functions that the liver cell, if they were there, could do. So it causes a bunch of dysfunction. And then add to that it can move to any other place in the body, and that's where you start seeing full systemic problems. And once they seed to that new area, they're not going to stop growing there. They're going to create other tumors, and then this will just continue to happen. So these tumors are abnormal masses of tissues. Cancer cells divide out of control, form these mass tumors, and then these tumors can either be contained, be considered benign, or malignant. Now, how do you remove or kill these abnormal cells? Well, like I said, you can surgically remove them, and it's very easy if they're benign tumors because they're not spreading to other parts of your body. You can also get anti-cancerous drugs that can slow or block cell division. The problem with these is that a lot of them will kill your normal cells too. So it's a nice balance between giving you enough of these anti-cancer drugs to kill your cancerous cells, but not kill you. Some cells are very sensitive to these drugs, and that's why you lose your hair. Those are some of the most sensitive cells that are in your body, or the ones that are at the top of your hair that head that provide growth to your hair. So that's why you lose your hair. And when you stop taking these drugs, of course, your hair grows back. Radiation also can kill and target cells. Again, they're going to be as precise as they can and try to give you just radiation to that area, but sometimes it's going to damage cells around it. For instance, my mom had squamous cell carcinoma, which is cancer of the mouth. And so when they were able to actually diagnose it, they started both chemotherapy, which are the anti-cancer drugs. She, of course, lost all of her hair. She got really sick because your digestive cells are very particular against it too. And so she would throw up and she'd have those. She got very tired because it is a poison that you're putting in your body. She also did radiation inside her mouth. And so though they tried to be very, very specific with it, it did end up killing a lot of the cells in her mouth. So she had problems creating saliva, and she also changed her taste buds radically, and she couldn't taste some things. So realize that it's going to affect not just cancer cells, but your everyday cells too. So will we find a cure for cancer? So it's one of the major discussions that we're talking about in this particular topic. So you'll want to look at the video below and get some ideas if you think we will find a cure for cancer. So a big part of protecting yourself from cancer risk or reducing your cancer risk is to actually protect yourself from cell mutations that develop in your genes that control cell division. So some of the risk factors that they note and ways to reduce them, a risk factor is eating on a healthy diet. So eating less saturated fats and more fruits and vegetables have proven to reduce your risk for cancer. There's things that you have to watch. For instance, if you really like some of the charcuterie boards that you eat, where it's kind of some of the processed meats, you want to make sure that they're not processing them with different forms of nitrogens, because kind of nitrites and nitrates can essentially have been linked to cancer. Obesity, so maintaining a healthy body weight and getting regular vigorous exercise can help. Tobacco use, quitting smoking or chewing tobacco, actually never starting, and even things like hookah or doing the vapes, you have to be very careful because these are not regulated as well as cigarettes are. So that's why I tell people, you think that they're healthy for you, but they're not. And a lot of times they're adding stuff to those combinations that can actually make them more toxic than cigarette smoke. So keep that in mind as you move forward in your life. There are a lot of environmental toxins that increase your risks. So minimizing your exposure to harmful chemicals both at home and at work. Protecting yourself with ultraviolet radiation. So not using tanning beds and actually putting on protectant to go out into the sunlight. There's some indication that some of the SPF that you use can be linked to cancer too. So some of the ways that you can do without using those kind of chemicals is to wear hats and cover your body. If you go outside in the summer, long sleeve shirts that are white and cotton can help a lot. There are cancer-causing viruses, so using condoms to avoid sexually transmitted diseases. HPV, for instance, is associated with cervical cancer, and aka genital cancer, but it's not just there. You can also get it in the anus and in throat. So this is something you're not going to be able to tell that you have. Getting regular pap smears can help protect you, especially if you are extremely sexually active and you're not using protection. And men, don't think that you're safe because you don't have a cervix. You can actually, and there's been chances of you getting it in both your throat, your mouth, and anus. Being able to test is very protective against cancers. So detection time, if you can't be diagnosed with cancers early in the process of developing it, you have more of a chance of being able to recover from them. So using self-test and medical examinations for early detection. Also knowing your family history and aging can help you, because as you age, or if you have a family history of things like breast cancer, making sure that you go and get routine mammograms to avoid late detection is what's important. And so just know that some mutations are inherited, but many are caused by DNA damage from diet, chemicals, and viruses. And so there are really cool things. And so there is a race against cancer. And this is what makes it harder to battle different cancers, because not every cancer is exactly the same. They work different. But they have noticed that there are some things that every tumor can be susceptible to. So they do have a mix of, when you have a tumor, there is a mix of drug-resistant and susceptible cells that compete for space for nutrients against your regular cells, right? So chemotherapy, what we figured out, is going to kill the susceptible cells. And what they see here is that there's a change of the balance of power. With these susceptible cells killed, these drug-resistant cells are freed from the competition, and they are able to divide more rapidly. So this can be bad, because if they're resistant to the chemotherapy drugs killed, then as you go through chemotherapy, they will not die. So what they're doing now is they're applying an adapted therapy approach to cancer. And so as you can see here, what's happening is the green cells are the susceptible cells and the red cells are the resistant. And in the original tumor, there are just a limited number of cells. But what they're realizing is this is the total volume and this is the days after the tumor ejection. So when the standard treatment, they have just the standard treatment where they give either the same dose or increased doses that they can give you. And then here, that's when they stop it. That standard treatment ends. And so you can see at around 40 days or so, they're all around the same number of cells. But the standard treatment, you can see the number of cells increase here, and they are the drug-resistant cells here, right? Now, they've also done where they looked and they skipped some doses. And so what they found here is that if you notice, there's less number of cells that are grown. And what they're starting to see is that, so if you skip a dose, right, that it's less, you're killing less of those original susceptible cells, so less cells grow back. Now, what's really interesting is when they start with a high dose of these drugs and then decline the dose as you go through. What you'll notice is that you have this low baseline of growth that happens. So what's happening here is some of those susceptible cells remain, and they're competing against the drug-resistant cells. The green one are the susceptible, right? And so you have less of the drug-resistant cells that form, and overall less of the tumor comes back. And so this is a dose, and you just keep kind of doing this in cycles until you're able to kill them all out. So this adapted therapy long term, because what you're doing is you're starting with less cells at the beginning of every treatment cycle. And this has very promising results. So we are done with, and so here's the last you can look. And basically, scientists have found an accidental way to treat all cancer cells. So this could talk a little bit about overall cure for cancer. And so we are finished with this overall lecture. Will we ever cure cancer? It is a group of diseases that we may never be able to cure completely. But as you know, scientists are very optimistic that vaccines, personalized medicine, and smart lifestyle choices will help to prevent many cancers and hopefully treat a much greater portion of cases than currently happens. So we are done with this section, and I hope you've enjoyed it. And if you have any questions, please let me know.